Okay, so cell membranes, individual cells, right, including the cells like nerve cells that we were talking about, are bound by a membrane. And that membrane, in a neuron, we're going to see is quite remarkable because it has all these kind of membranous projections, the dendrites, these branch-like projections off the cell body, the axon, the wire-like extension right here, these axon, what they're called collaterals that sort of come off of the, into the main axon here, and then the terminals, the very end points here, and the whole structure is bound by what's called a phospholipid bilayer, a membrane, a cell membrane. And it's called a phospholipid bilayer because one of the primary chemical constituents, that make up this this membrane is something called, it's a class of molecules called phospholipids. And these phospholipids are they're illustrated by these little ball and stick kind of images here. They basically have a charged phosphate head. It's negatively charged. And charged substances like other charged substances. So the phosphate end of this, the head end of this molecule, will like to be in charged environments, like watery environments. H2O has a positive and a negative end. And inside the cell, on the intracellular side, there's lots of charged substances, water, ions, all sorts of things. And so the phosphate head's going to want to orient intracellularly, and then also on the extracellular, outside the cell, is lots of charged material, charged chemicals, charged stuff, water and other things. So the head's going to want to orient to the extracellular side. But these molecules, phospholipids, not only have this negatively charged phosphate head that likes charged environments, intracellular and extracellular, but they also have this long sort of lipid or fatty tail, which is not charged. And things that are not charged like to stay in environments where there are things that are not charged. So these tails tend to kind of cluster together. So they have this, the membrane has the phospholipid, the phosphate heads, oriented extracellularly and oriented intracellularly. And then in between is this layering of lipid tails. Hydrophobic. They don't like water, right? They don't like charged substances. So there's like this fatty internal, aspect of the phospholipid bilayer. And it's a bilayer because here you've got one, row of the phosphate heads with the lipid tails. And then here you've got another row with the phosphate, heads and the lipid tails. So you've got this bilayer that surrounds, that forms the entire membrane, of cells. And this is not just nerve cells, this is all cells basically in your body. And one of the useful things about phospholipid bilayers is that they are largely impermeable to the passage of charged substances. So we're going to see that it's very important that neurons maintain differences in the charges, the different kinds of ions. We're going to see these are atoms that are missing an electron or have extra electrons. So they're going to have a charge associated with them. Well, those kinds of things can't easily cross those membranes. And in addition, there are many chemicals that are going to only be able to interact with the cell membrane, externally in the extracellular space because the membrane isn't permeable to the passage of those chemicals. Like, for example, a neurotransmitter we're going to see is going to act at machines that are embedded in. These are proteins embedded in the phospholipid bilayer, but they can't actually cross the bilayer itself. And also, it's going to lead to a very significant difference in terms of what chemical constituents do you find intracellularly inside the cell versus extracellularly, because things that are manufactured, like proteins that are built inside cells, can't easily escape them if you've got this, fatty internal, lipid portion of the phospholipid bilayer that prohibits the passage of charged substances. We're going to see other kinds of chemicals, like, for example, alcohol is a chemical that can easily pass through, phospholipid bilayers. And some chemicals can more easily cross if they're more lipid in nature. We'll see cannabinoids, for example, can easily cross membranes as well. Now, the other thing I want to mention here is that In order to get substances from one side of a phospholipid bilayer to another, which is largely impermeable, the cell needs to embed protein machines in that phospholipid bilayer. And there are a number of different kinds of critical protein machines that can be embedded there. There are, for example, receptors. And we're going to see at synapses where one neuron meets the other across a gap called a synaptic cleft or a synaptic gap. They're going to be embedded in the membranes, both presynaptic, the sending neuron, and postsynaptic, the receiving neuron. You're going to see like a lot of big proteins that are kind of embedded in the membrane that span the phospholipid bilayer, and they have on the extracellular component like some kind of a binding site where the neurotransmitter, let's say, that's released from the first neuron, the presynaptic cell, can bind, and so that receptor can receive that chemical message. And we'll see what it does is it'll change its shape in some fashion that will alter its function. And then, one very important thing to keep in mind about receptors is typically they're going to bind, like let's say a neurotransmitter, they're going to change their shape, and then they're going to release the neurotransmitter and change back. So there's a temporal or a time component of this. There's interactions. These are drug actions, right, on receptors. And they can be sort of limited. Drugs, we can see, can also act often at these receptors. They can, block their action. This is called an antagonist action, or they can actually act just like a neurotransmitter and cause the change and, cause the thing that would normally happen with a neurotransmitter. That's called an agonist action. We'll talk more about this later. Another kind of protein machine that can be embedded in the membrane, in the phospholipid bilayer that surrounds all these cells is something called a channel. And a channel protein is what it sounds like. It's got a channel. It has a hole that goes from one side of the membrane to another. And this hole usually is selective. It can only let in or out, a particular ion, like a charged particle, like a sodium ion or a chloride ion. These channels, by the way, can also be open all the time. They call these non-gated channels. They're in a structural shape or conformation that allows for the passage of ions in the direction that natural forces will drive them at any time because they're just open all the time. While others are actually what we call gated. They have more than one shape, more than one conformational state, more than one structural kind of form. So they can be gated. That means they're closed. There's no hole. There's no pore. There's no open channel to allow anything to go through. Or they can be ungated. They can open, right? And what gates them, we're going to see, is going to be very interesting in the context of a course on drugs. Drugs, for example, might be able to ungate, a gated channel and alter the charge difference across the membrane, alter the distribution of ions across a cell's phospholipid bilayer. So we'll talk a lot about what gates these various channels and can cause changes in both structure and thus the function of these channels under certain circumstances. Now there are also, and another important kind of protein machine that can be embedded in the phospholipid bilayer of cells, including neurons, is something called a transporter. And a transporter literally transports something from one side of a membrane, one side of a phospholipid bilayer, across to the other side. But it's transporting. The word that's implied here is actively transporting. It's fighting some kind of a force, like the thing that it's grabbing onto. This is a protein again, right? That's built by your cells, called a transporter, embedded in a membrane, embedded in a phospholipid bilayer, that's latching onto something on one side of a membrane and forcing it into the other. It's fighting, various forces. These chemicals that it's pushing, for example, from one side to the other wouldn't normally go there. They would normally sort of want to go the other direction. And that's because there are a number of forces that are, playing a role here, including something called the concentration gradient force, which basically states that, substances that are in high concentration tend to move towards areas of lower concentration. So if you want to get something packed in high concentration on one side of a phospholipid bilayer or a membrane, you're going to need to fight that concentration gradient force. and you're going to need to put in energy to make this effective. And the form of energy that cells use is something called ATP. It's a molecule. It's generated from the burning of glucose, burning of sugar in the presence of oxygen in these little cellular organelles called mitochondria that are kind of like the energy powerhouses of the cell. And that's why you need to eat and breathe so you can keep generating ATP. And there's chemical energy in those bonds. And so what a transporter does is, it's sitting there in a membrane, a protein spanning the phospholipid bilayer. It grabs onto something, say, on the outside, right? And then it'll change its shape, right? And that change in shape will allow for ATP to fit in and a bond to be broken. And that chemical energy is going to be then transferred into the kinetic movement or transport of the chemical from one side of the membrane to the other. So the energy is, neither created or destroyed, but it's being converted, from one form to another, in this case, from the breaking of the chemical bond into the kinetic, transport of a substance from one side of a membrane to another against some force. So a transporter is a really important kind of molecule protein machine that we're going to see in phospholipid bilayers at the synapse. There are, you might have heard of selective serotonin reuptake transporters. There are selective dopamine reuptake transporters. There'll be a lot of protein machines that'll grab onto, let's say, a neurotransmitter that has been released into the synapse and then pull it back in. And it's got to sort of fight forces to do that. It utilizes ATP. So that's a transporter. We're going to talk a lot about transporters. And the final kind of class of protein machine I'd like to mention is something called enzymes. So these are proteins that are also expressed by genes, transcribed and translated. And then they can grab onto, for example, let's say 2 raw materials. And then because they grab them both together, they stick them together and they form something new. And we're going to see when we're synthesizing, or building neurotransmitters, there's often what they call a synthesizing enzyme or a series of synthesizing enzymes that'll, grab the various raw materials, stick them together, create this, chemical. Then maybe another enzyme grabs that and does this to it, and then, builds, for example, dopamine or norepinephrine or one of the neurotransmitters. So synthesizing enzymes are critically important. And also, we're going to see the chemical messages that are being, sent at synapses are time limited. And there's typically a process of degradation, of breaking down the neurotransmitter, right? And these are out there. These are also, it's often because of enzymes. So it'll often be like an enzyme will grab onto like something and then rip it apart, degrade it. These are called degradative enzymes. So receptors, channels, both gated and ungated, transporters utilizing energy in the form of ATP, and enzymes which build things up, synthesizing, or break things down, degradative, are 4 important classes of protein machine that we find in this phospholipid bilayer, embedded in these phospholipid bilayers. And these are not only the targets often of neurotransmitters, but also we're going to see of drugs.